Thank you, Tiago and Dayo for the invitation. Yeah, I think you guys are going to get an overload or overdose of VMH. I think right, so you'll see, and I hope, if not just during the talk, but that later we can discuss several aspects of what I'm going to talk about, and in relationship to what Dayo already talked, because the main difference is that I work on the female VMH-VL, and there's a background for the female VMH-VL that is quite different from the male VMH-VL. But I think already with Dayo's work and with some stuff that I'm going to show you today, I think we are kind of converging more and more, maybe that the structures are actually not so, there's more similarities than actually differences. So yeah, so I started I did my PhD with Gero Miesenbach in a time where optogenetics didn't exist. That makes me feel really, really old, but yeah, so I was pretty lucky to finish my PhD work before Diserov's paper came out, and so essentially what I did during my PhD was to make flies fly artificially. So that's what I did. That's what I did at work with Gero. But essentially, it was the first demonstration of this powerful field that we have today, optogenetics. It was not as fancy as the ones, and not as useful as the ones that we have today. But it was the first demonstration of the ability of controlling animal behavior by light, and by controlling directly the conductances of some channels in some cells. Then I moved on to Cold Spring Harbor, and there I did a bunch of things, but primarily the thing that ended up coming up as a paper that I already mentioned was PIMP. I was very proud to also come up with that name. So it was this so it's using channel rhodopsin. So everybody was using channel rhodopsin, or most of us use channel rhodopsin to activate cells and to try to find a causality arrow between neural activity and behavior. And in this case, what we had the idea was to going back, so most of us do recordings, and if you're doing electrophysiological recordings and you don't know what your cells are, so you imagine independently if you are in the VMH, you have cells that express estrogen receptor, cells that express leptin receptor. So you have a zoo of molecular markers and different cell types in every cell, in every region that you record from, and rarely you are able to put a name, an identity on the cells that you are recording from. So the idea was to pre-select some neuronal population from the brain region that you are recording from, for example, the estrogen receptor population in the VMH, and put channel rhodopsin on those cells. And then you do, your animal is doing your behavior, you record the activity of those cells, and in the end, you just ask a very simple question. Is any of the spikes that I'm recording during my behavior coming from an estrogen receptor positive cell? And you shine light over the recorded area, and you, the spikes that you see that are light evoked, you know that they are, there are some tricks to make sure that the spikes that you are recording are coming from the cells that you are directly activating, and then you can match that to the spikes that you recorded during your behavior, and so you have one extra piece of information, so not only how the identity of the cells, but how those cells performed or behaved during the paradigm that you were studying. So since then, I started my lab at the Champalimaud Foundation. I think some of you, there's some people here that came from there. I have one of my postdocs here in the school. And so since I started my lab, I've been working on I completely switched fields, and I think I was like a late bloomer in the sense that I'm interested in many different things, and I think it took me some time to understand what I really wanted to study. And I think I think at this moment I'm really happy with what I'm studying and and I think that's what I'll be doing for the next few decades. But if I actually think about how did I get to the interest that I have today They go actually very far back to the beginning of my scientific career. And I'm going to show you just a little movie that has nothing to do with the work that I do, but I think it was one of the first times that I got really, really interested in sex, okay? I'll just leave it like that. Okay, so I don't know if you ever saw hedgehogs mating. So when I was doing my Erasmus program in Holland, one of the nights, I don't know, I remember like coming out from downtown Amsterdam and going to my dorm, and I was like cycling. I don't know. It actually like I think there was like a little bit of snow, and no, it shouldn't be because it was no, it doesn't matter. So anyway, so I was cycling, and suddenly I start hearing like a really, really, really loud noise, which I had no idea what it was at the moment. But then I went, like I got out of my bicycle, I went to see what was going on. And so this is not the movie from that time, but this is just from the web. So this is the male and the female hedgehog. And I don't know if you ever actually saw them mating, so, or if you ever played with a hedgehog. Okay, so like hedgehogs are really scared animals, and as soon as you try to touch them, they will curl so that they can protect themselves. In contrast, during the mating season, these guys will just be, so the ones that I found, they were in the middle of like in the middle of, it was an open field, like everybody could spot them. They were making this incredible noise. I stopped, I went to see, like I spent, I don't know how many minutes looking at them, doing all this sniffing behavior and then mating. And I was like really amazed by the fact that how is it that this animal that are, like incredibly shy, that will run away and hide at the first sight of a predator, which would be me, in this situation, like there must be such an internal state, like such a drive, that this animal, like even though I'm there and like they are in the open field, they keep on doing this behavior and they are not perturbed. And I can tell you many experiments that we do in the lab nowadays where we do things that I never thought that we would be able to do to the animals while they are performing sexual behavior, which I think we don't understand what are the neuromechanisms that maintain the animals performing these behaviors once they start, but I think this is one of the fundamental questions. So for example, one of the things that we do is that we take blood from animals as long as they perform sexual behavior in order to measure the level of hormones in circulation. And so what we do is that we have the animals performing the behavior, and every 20 minutes we pick the animals, we do a little cut in the tail, we draw blood, and then we put the animal back in the box, and they keep on performing the behavior almost the same way as if you didn't disturb them. So there's some very, very strong mechanism that ensures that once you start the behavior, you actually take it to completion. So that's one side of the thing that I find it particularly interesting. The other side is actually like, if you think about sexual behavior, and this is another aspect that we also don't really understand how this works, is sometimes people ask me, like, do you study social behavior or not? And I say, like, sexual behavior, I don't necessarily see it as a social behavior in the sense that you have like many animals that are asocial and that live in isolation, but that during the time of mating, they actually need to be in contact with each other. So it's not the classical type of social behavior. Animals do things together in order to, like they do something together in order to achieve a goal, but it does not necessarily need that the animal that is performing sexual behavior is a social, so-called a social species. And if I think like I have two kids, if I think about sexual behavior, or for example, maternal behavior, is one of the two of the strongest examples where my own identity, my own, so my boundaries have to be this almost like dissolved in order to allow this other social, this conspecific, in order to get as close to me and in a situation where there's a very strong drive that allows for these behaviors to happen. Okay, so and there's a lot of theories around there relating like the vagal theory of social behavior. There's a bunch of things that I think are interesting to discuss, but for me, those are the two very interesting parts of the behavior. One is that you have to, and I think this touches very much on like defensive behaviors, on aggressive behaviors, that you have to inhibit and tone down these behaviors in order to allow a conspecific to come as close to you, to be on top of you, and to allow you to be in a situation that is incredible. It's defenseless. You are like exposed to predators, for example, and that once the behavior starts, you ensure that like there's mechanisms, nature selected mechanisms that allow you that you go all the way to the end. Okay, so that is kind of like my personal interest in the behavior, even though like I'm interested in these higher level questions, and most of the things that we do in the lab, they are just like very reductionist approaches or intents to get to these higher level questions. Okay, unfortunately, or maybe fortunately, we don't study hedgehogs, we study mice, and let me just show you a little bit. I think most of you probably already seen how like mating occurs in mice. So usually what you have is a period of mutual investigation, and during this period of mutual investigation, animals are trying to gather information about each other. And like I will not go much into detail about this, but it's that like you can get a lot of information. These experiments, our experiments are usually done in the dark, even though we can under red light conditions, so we cannot rule out the possibility that they are using some visual cues. And I think one of the things that we are also very interested in is understanding if the VMH VL is also receiving visual and auditory cues, which could allow for females, for example, to understand the gender, the status of the animals that they are interacting with. But most of the action happens at the olfactory level, and so animals sniff each other a lot in the facial region, but primarily in the anogenital region, where there's a lot of cues that are important for conveying information. So if things go well, and there's many aspects that, or many factors that will allow the next phase, the consummatory phase, to happen, which I'll discuss later. At some point, the male starts trying to mount the female. And this usually, at least in laboratory conditions, and we don't know, we know a little bit how things happen in the wild, but we are not, like, we don't have a full idea of what actually happens in the burrows when animals are mating or outside. But usually, at least in laboratory conditions, what happens is that the male at some point starts mounting the female. And so what we call these mount attempts. And I think I'm going to switch to this one. And during this phase, what happens is that the male tries to, it chases the female, it tries to put the forepaws on the flanks of the female. And again, in laboratory conditions, what happens is that the male usually starts this behavior, and the female initially, even if she's sexually receptive, she will reject and she will walk away from the male. It's really, really rare that at the first instance that the male tries to mount with the female, that she will accept him. But as things progress, females will allow the male to mount them. And actually, before we go, so at some point, what you have, what we call mounts with intromission, that at some point will lead to ejaculation. So sex in mice happens in a very particular way. So you have several bouts of mounts with intromissions, and then these mounts with intromissions, these mounts. And this part of the behavior, even though the consummatory phase is highly stereotypical, the number of bouts of mounts that you can have is incredibly variable across animals and within the same animal. So sometimes, and at this moment, we did like hundreds of experiments by now, and we cannot predict, we cannot say what, like when we put them together, what's going to happen, okay? There's nothing in the behavior of our male mice or the female mice that can tell us like how they are going to behave. So sometimes you put a male inside a box, and within 2 mounts he ejaculates. Sometimes it takes 30 mounts for the animal to ejaculate, the same animal, okay? So I'm not going to talk about this, but one study that we are finishing at this moment is looking at 10 couples over 10 sessions and trying to understand if there's, yeah.
So why would males not just ejaculate immediately?
So that's a very interesting question. And for example, this is very different from rat behavior.
I mean all males.
Sorry?
I mean ubiquitously, right? You're in this vulnerable, as you were mentioning, you're in this vulnerable.
Yeah. So there's a lot of speculation in the field. And again, I don't have any serious or whatever. I don't have any data-supported answer to you or a final answer. Or I have data-supported answer, but not a final answer. In the sense that so the way we do our experiments, the males are trained before. And for everybody that works with black six, you know how painful it is to work with these animals, even for sexual behavior. They only perform once a week at maximum, and sometimes even not once a week. So it's really, really hard to do experiments with these guys. We also work with wild-derived animals, and those guys, they will have sex. 40 minutes later, they are having sex again, and they can have more than once in a day, and they will do it several times a week. So I don't think these guys are also a very good model, but even those wild-derived animals, they take several months in order to ejaculate. So.
But you have some ways to increase their sexual behavior.
So you could say, okay, so some people say it's from the female side, so you need enough vaginal stimulation in order for pregnancy to occur. So there's some really crazy and interesting studies that were done. You actually don't need a penis, you don't need a male in order to make the female pseudo-pregnant. You just need mechanical stimulation of the reproductive organs. There's some studies that were done just, in inflatable balloons that you can put inside the vagina, and you can cause pseudo-pregnancy with that. So there's some people that talk about that. So we have some data looking at, so we are recording in the VTA while males are having sexual behavior. And we are also now doing very, very fine characterization and annotation of the behavior. And as far as we tried everything, or a bunch of things so far, we cannot find anything that predicts what the behavior is. So the duration of the mounts is variable, the number of the mounts is variable, and it's variable across animals. The only thing that we know is that across time, they seem to take less time in order to initiate the behavior, and they seem to need less mounts in order to ejaculate. But it's also not it's not a huge difference. It's not that the first time that they have sex, they need 30 mounts, and then the last one, after 10 sessions, they only need 5. And it's not the same across all animals. So there's the only thing I can tell you is that it's incredibly variable. We don't know what is triggering ejaculation, for example, at this moment. This is one of the things that we are trying to investigate. So we don't know if the behavior, what they are doing, we assume that the behavior is necessary in order for ejaculation to occur, but we don't know, it could be that every mount or every intermission is different. And some of them are done the proper way. Others are not done the proper way. Some people say that this is important for female. This is called also pacing behavior from the female side. So we don't really know. Okay. Yeah.
Under what condition? Those animals are usually under not test condition. Are they together? Are they? Those male and female, do they know each other before? So
we have all sorts of different experiments. Okay, so the males usually, once they start having sex, you have to isolate them. Otherwise, they start becoming quite aggressive to each other. So the males tend to be isolated.
From males or from females?
From other males, and they are not with females. They are not together with the females. So we control when they are put together. Okay, so they are not always together. The females are in in groups of like 4 or 5 females. Another thing that I think is very important, and this, I think it's a huge controversy in the field, is that different labs do experiments in different ways. So many labs do OVX and then hormonal priming. And females in those conditions behave completely different from females that are naturally cycling. And so we use both types of females. So when we want to study the female, we use naturally cycling females. When we want to study the male, we use OVX females plus priming. But I think it actually really changes also the way. So if you have a naturally cycling female, the male is mounting her, and usually the female runs away at some point. If you have an OVX primed female, what happens is the male is mounting, and at some point he stops, and the female still stays there. And then like you look at them and it feels like, oh, like I'm still on top of the female, and then they start doing the behavior again. So it's actually quite different, and so we are also like investigating that. Okay, so I think we also need to be careful regarding that aspect.
Do you also observe some called Coolidge effect when the motivation to engage in sexual behavior depends on the novelty of female life?
So we never tested that. So we studied the refractory period, but in those cases, so like how long does it take for the male to regain interest, sexual motivation after ejaculation? But in those cases, we kept the female the same. Okay, we didn't. It's something that we would like to try, but for most of our studies, when we give another female, is the same one that you already had sex with, because females can have sex several times, also. Okay.
Because you have no way to say if anyone is happy or not.
Yeah.
I'm wondering if the facial and this is maybe the best way to see if facial expression here changes, and then you can like transfer it to other behaviors. If you have some other behaviors, you think anyone is happy or not with the same muscle right? Do you have recorded?
They definitely. Or are you asking if they like the behavior?
No.
Or
if you have any indication if the animal is happy, like facial expression.
No.
What you've tried doing to from face?
No. That's interesting. Yeah. Yes.
Did you look at the hormonal cycle in the female and how it affects the
Okay, we'll get into that. That's the main part of the talk. Okay. So just to again, just to see one of the things that again that we also are very interested in studying. If you look at the male behavior, so it's really easy to identify when the male ejaculates. He essentially drops to the side. He becomes immobile and he's panting. And then at some point, what happens is that he gets up, and many times he goes to the corner of the cage. He does genital grooming, and he rarely, for a variable period of time, he doesn't want to have more sexual interaction with the female. And many times the female is going there and trying and poking him, but he rejects her. Okay, so I'll just leave it like that again. Okay, so I just added this slide while in the previous talk because of Sam's question. And so many people, I already taught like behavior at university, like undergraduate students, and I mean, people ask, like, but why do animals have sex? Like, why do we have sex? And like, what is the, can we think about, this is not going to be a very deep discussion, but I think like I'm just bringing this up because I see that there's some questions there in the table, and that I think this is an interesting topic to discuss. So I think we all agree that regarding sex, it's not just drive. So is there a need, and like do animals like just perform in order to do drive to do drive the reduction? I think we all agree that in sex it's quite more complicated than that. But animals will not die if they don't have sex. Like one of the questions is like, are animals actually less happy if they have sex? It's like, again, if most of you, like for most of you that do experiments with black 6, if you try to see, there's always a percentage of animals that actually has absolutely no interest in sexual behavior. there's 10 to 20% of these animals that you put them together with a female and they will never engage in sexual behavior. They just pet the female, but they don't engage in sexual behavior.
Does that matter if the male is like very dominant male?
Is it what, sir?
Like in hierarchy.
We didn't look into that. Like, it's a variable that we are not, we try to match, so we have the age matching and we have the weight match, but we don't know anything about their social status, even though they are social isolated. So we hope to get rid of most of the the dominance.
In nature, at least, the males they fight, and whoever is more dominant goes for the female.
Yes,
It's
even though
those males that they have less sex, they are less dominant. If you look at the hierarchy and pick up the more strong male, you will have more frequent sex.
Yeah, I don't know. Okay, we will try to. It's something to test. Okay, so and again, so these are just ideas that I think that we could bring into discussions. And so the way I think, and I think most of us think about sex, is that so there's some physiologically based, it's a physiologically based motive that so there's hormones that are changing and creating maybe what we can call needs, similarly to what we think about feeding, for example. But there's also a very strong component related to incentives and also to learning. So it's really, really hard to disentangle, even in humans and in mice, what is actually innately sexually rewarding. Many people say that only genital grooming is the only thing that is innately rewarding, and that everything else is learned by association during the consummatory act. And obviously in humans, there's a lot of social values and other stuff that make things a bit more complicated. So, but anyway, so I think it could be interesting to discuss these aspects. So one way that we can look at sexual behavior in a more pragmatic way is that so we have what we call, so the initial phase, we have the appetitive phase, which is essentially it's a set of behavioral of actions that will allow the animal to be in contact with the thing that the animal wants to consume, which in this case would be mounting or so lordosis from the female. And then at some point what we have is what we call the inhibitory phase, which is important also to stop the animal from making that behavior, and that allows the animal to go and do other stuff. And so this inhibitory phase, which we also study, we know that it acts both on the appetitive circuitry and the consummatory circuitry in order to ensure that the animal, once he reaches the end of the behavior, things stop. So we work on a bunch of different things, and if anybody has any particular interest in any of these topics, we can discuss during this afternoon. We work on mate choice, and we have very interesting data on mate choice. We also work, as I just mentioned, on VTA in males, and we are trying to understand what is the role of VTA during consummatory behavior primarily. We are doing something that I'm very excited about, which is we are studying ejaculation and the refractory period in the spinal cord, and this is something that Constance, that is here, it's part of her project. We are trying to understand what are the spinal circuits that control ejaculation, and one of the main reasons for doing that is because we would like to understand what happens at the point of ejaculation and what are the changes that happen that allow that animal to enter this refractory period. Again, one of the things that I find fascinating is that seconds before the animal ejaculated, this female is the most rewarding and amazing stimulus in the world, and immediately after ejaculation, it doesn't want to interact with her anymore in a sexual manner. So something really profound and fast happens in the male brain that this thing changes. And also, again, related to this idea, we've been studying for many years, we've been studying prolactin. And again, if anybody has any interest in this molecule, there's like a long, decades-old idea that prolactin is one of the main players in the setting of the refractory period. And through a series of quite painful experiments, we think we can really show that prolactin is really not necessary or sufficient for the refractory period. So like if anybody is interested in discussing. It's only negative data, but they are very cool experiments, and I think we are going to finally close a chapter that has been open for many years. So what I'm going to talk about today is about receptivity and the hypothalamus, and again, I'm going to take you back to the VMH, and touch on many things that I already mentioned, but from a different perspective. So one of the things that is really cool is that females, this is very loud. So this is a female and a male. So this is quite loud. So this is a male and a female, and this female is not receptive. So the female is here, the male is going to drop from the roof. And as you'll see, this female, she's like in the upright position, and every time that the male tries to talk with her, she pushes him away. And in contrast, 3 days later, the same exact couple will have a very different behavioral output. Okay. So the female essentially goes into what is called the lordosis posture, and when the male touches her flanks, is able to go on top of her and copulate. So this is one of the questions that we are interested in, is understanding how the same stimulus, the male, can drive such different behavioral outputs in the same animal, depending on the internal state. So briefly, because I think we all come from different backgrounds. Yeah.
On purpose, you have random noise always?
I have what, sorry?
You have random noise always? Or does it perform the experiment? You have random noise on purpose?
Oh, this is the noise from them actually making the. So there's part of the noise that comes from their cage, and also we do our experiments in the same room where the animals are kept. So we had to decide, so actually in the beginning we didn't have enough space, so we could only do experiments in the same place where the animals were held. So we do everything in the same place where the animals are held. We also don't have ventilated cages, they are standalone, and so they are exposed to, like, they know everything about like whoever else is in the room from volatiles, okay? Okay, so what drives these changes or what drives the reproductive cycle? So the hypothalamus is the main controller of the reproductive cycle. So there are neurons within the middle prooptic area which release hormones that are going to be released of the GnRH, which are released into the pituitary, and in the pituitary there's going to be some hormones, the luteinizing hormone or the follicle stimulating hormone, which are going to be then sent into the ovaries, and then it's ovaries that make progesterone and estrogen, and these now the so-called female sex hormones are the ones that then are going to control a variety of different alterations, like physiological changes, but also are the ones that are going back to the brain and can change the way circuits work. So this HPG axis leads to the beautifully orchestrated reproductive cycle. So this is the human menstrual cycle where you have, it doesn't really matter like the different lines, there are different hormones. This is the rat and the mouse is kind of similar, but essentially what you have is that through the action of the hypothalamus, the pituitary and the reproductive organs, you have varying concentrations of hormones across different days. And so it's quite incredible that you have a signal that is varying over time, and then the question is like, where is this signal able? So this signal can bind to many things in the body, but it can also bind to many things in the brain. And so many people ask me like, where are the receptors for these hormones in the brain? Are they concentrated in very specific regions like the hypothalamus, or are more they're widespread? And so many people get very surprised when they see a picture like this. So this is an in situ, so this is just taken from the Allen brain, and this is the in situ for progesterone receptor. And I think you can appreciate that, at least in this case, and for estrogen is a very similar thing, that scenario that these receptors are expressed like widely throughout the brain, like in the cortex, in the thalamus, in the hypothalamus. I think like the cerebellum is one of the most spared structures, but essentially, like, so we have this varying signal and we have receptors that are expressed all over the brain. So what are these molecules doing to neurons? They do a bunch of things. They have genomic and non-genomic actions. Yeah.
Bit of an esoteric question, but so release of reproductive hormones is ultimately controlled by the brain.
Mm-hmm.
So could there be any role for neuron, the brain directly influencing internal states related to reproduction, rather than going via reproductive hormones that do LHRH neurons have some
yes,
collaterals in the brain?
Yes. Yeah. It's kind of a I don't know. It's a mess. It's a beautiful mess because these neurons they control they can project to they release hormones that can control the pituitary, for example, but they also work as regular neurons, and they have postsynaptic targets that are involved in the control, the social brain network of a bunch of different things. So yes, and also there's something that we tend to ignore, which is these hormones can also be produced in in organs that are not reproductive organs, including in the brain, for example, like prolactin is produced in the brain. So it's complicated to understand exactly what the signal is many times, because just doing systemic manipulations of these hormones doesn't address you, for example, some of these questions that you are addressing. Okay.
You showed the allograin RNA expression of
progesterone receptor
in adults.
Mm-hmm.
Do they also exist in P10, P20? This much of expression of receptors?
I don't know. I don't know during development. I know that they are there, but I don't know how it looks through development. Okay, so what do these things? So they have genomic and non-genomic actions, and so what are some of the things that they can do to neurons? So one of the things that estrogen and progesterone, for example, do is that they can change the expression of biosynthetic enzymes for neurotransmitters, for example. We know that production of dopamine, for example, is affected by the estrous state. So the amount of dopamine that females produce, for example, changes across the reproductive cycle. We also know that the expression of receptors is affected by the varying levels of sex hormones. And another thing that I find quite interesting is that even the structural properties of neurons can change through the estrous cycle. And so this is a very crude experiment that was done in the 70s, where it's not the best experiment, but I think at least it shows the potential of what might be happening, and we already have some data that is supporting these structural changes. So in this case, we're looking at the preoptic area, and we are looking at neurons from animals that were OVX, so they have no circulating hormones. And then they were treated with estradiol and looking at the dendritic hybridization of these neurons. So what we know at this moment is that the structure of neurons in different parts of the brain, particularly in the hypothalamus, and we are already seeing that in the VMH, is changing across the cycle. So you have extension and increase in the richness of dendrites across the cycle. So you have, for example, extension and retraction over a very short period of time. So of course, this is going to affect the way that circuits are going to lead to behavior. Okay, so I'm going to go faster because otherwise I'm not going to go through my talk. Okay, so sex hormones change. So behaviorally, what do the sex hormones do to animals? So we know that, for example, the attractiveness of the females can change through the cycle. These are experiments where males were just put into contact with females, either anestrus or estrus females, and here we're looking at the number of spontaneous erections in the presence of these females. And so males will have spontaneous erections more in the presence of estrus females. So there's something in the female that is different that causes spontaneous erections to occur, for example. We know that sex hormones can affect the way that social stimuli are perceived. For example, changing the detection thresholds of discrimination ability, and this has been shown in rats and humans. We know also that even in humans, our preferences for male faces can change across the cycle. So there are some studies showing that females prefer the